Lecture 2 is on the cell. In this lecture, some of it will be reviewed from your previous bio class, and then some topics will be new. What we're going to cover is an overview of cells, followed by the plasma membrane. Then we'll discuss the cytoplasm and the nucleus. And we'll end up with the cell life cycle. So you remember this figure from our first lecture, and we were talking about the levels of organization of the body. So today we're discussing the cellular level. So let's give an overview. First of all, cells in our body are numerous and diverse. Numerous meaning we have trillions of them, and diverse meaning there's a lot of different types of cells. All of these cells in our body have very specific structures and functions. For example, take a look at a sperm cell. Look at its structure with the head and the long tail, and we know that the function of the sperm is to help with reproduction. Let's look at another cell, red blood cells, which you'll see in lab. Red blood cells, if you take a look at them, they are circular, very different than the sperm cell. They're really small, and their function is to travel through all of your blood vessels to help deliver oxygen. Here's another cell, a nerve cell. You'll notice that it has a cell body right here and a very long extension. That long extension helps a nerve cell communicate to far parts of your body. And finally, let's look at a cheek cell. The cheek cell, if you notice, has a very irregular shape, but if you put a bunch of cheek cells next to each other, they all fit together perfectly like a puzzle to help form a protective barrier. So here's just 4 examples of 4 different cells, each with their own specific structure, which helps it serve its function. Like I said previously, we have numerous cells. We have about 50 to 100 trillion cells in the body. Trillion is basically 30 zeros. So that is quite a bit of cells. And the cells are diverse. We see 4 types here, but you have over 250 types in your body. Other cells which we haven't mentioned, things like skin cells, muscle cells, bone cells, and many more. And all of your cells, all 50 to 100 trillion, can be actually simply classified as either somatic or sex cells. So let me tell you what the sex cells are first. Sex cells are sperm for male or eggs for female. Now, all the other cells that aren't sperm or egg, those are considered somatic cells. So somatic cells are the remainder of the cells in your entire body. So now we're going to review the 3 main parts of a generalized cell. So you've probably seen this before in your basic biology class. So recall, every normal cell has a plasma membrane, which you can see right here, going around the cell. And its job is to basically separate the inside of the cell from the outside of the cell. Next, you have the cytoplasm. The cytoplasm is everything inside the plasma membrane. So this whole area inside is the cytoplasm. So within the cytoplasm, of course, we have many structures. We have the cytosol, we have several types of organelles, and we have inclusions. And then finally, we have the nucleus. And the nucleus tends to be towards the center of the cell, right here, and it is considered the control center. Now we're going to go into each of 3 of these parts in more detail. So let's start off with the plasma membrane. All right, the plasma membrane is considered a fluid mosaic model, and I'll explain why in just a little bit. Let's take a look and see what we have in the plasma membrane. So here, this is from figure 2.2 in your textbook, showing you a part of a plasma membrane. So if you take the plasma membrane, just this little section right here is blown up so that you can see. On the outside of the cell here we call the extracellular fluid. Extra means outside, so think X or exit. And most of this fluid is water. On the inside of the plasma membrane, you have the cytoplasm. Again, also mostly water. One of the main structures in the plasma membrane is your phospholipid bilayer. And you can see that right here. Here is one layer, and then right below it is a second layer. So that's where the word bi comes in. Bi means two, and these are all phospholipids. So there's two layers of phospholipids. Now, if we look into a little more detail in this phospholipid bilayer, you'll see that each phospholipid has two parts to it. A hydrophilic head, seen here on the outside, the little ball, and a hydrophobic tail, which are on the inside. So if we were to actually blow up the phospholipid bilayer and look at phospholipids, this is one single phospholipid. The head here is a circle, and the tail is right here. There's two of them. So now we look at the entire two layers, and it looks something like this. So you'll see the heads here, and then the tails facing each other on the inside. So let's see why. If you notice in front, we were calling the head a hydrophilic head. Now let's try and recall what hydrophilic means. Hydro, you've probably heard of that before. Hydro means water. Phil means to love. Not sure if you guys know, but there's a city called Philadelphia. And the reason why it's called Philadelphia is because it's got the word Phil. It's supposed to be the city of brotherly love. So that's where hydrophilic comes from. Water, love, or it loves water. So the hydrophilic heads love the water. So let's take a look. Notice all of the heads right here are facing the outside, the extracellular fluid, which is, again, water. So these heads are happy. They're facing water, which is what they love. Now, look on the inside. Remember, the cytoplasm is also mostly water. So here we have a bunch of hydrophilic heads, and they're facing the inside, which is great because, again, the heads love the water. But if we look at the phospholipid, here we have the tails. The tails are called hydrophobic. So let's review what that means. Okay, we just said hydro. That means water. And then phobic, or think of phobia. What does it mean to have a phobia? Well, it means to be afraid of something. So when we say hydrophobic, it means a fear or afraid of water. So the hydrophobic tails do not like water. So notice in the phospholipid bilayer, the tails all face each other on the inside, which means they're with each other, they're not facing water at all. And in fact, that's the reason why you have to have two layers. Imagine if you only had one layer of phospholipids, the heads would be on the outside, that's fine, facing water, but then the tail would be on the inside, facing the water of the cytoplasm. And again, the tails don't like water, so that wouldn't be a good setup. So having the bilayer makes sure that all the heads are facing water and happy, all the tails face each other and don't face any water. Now, this bilayer that I just described to you is fluid. So let's see why we call it fluid. Just imagine what a fluid is like. Think about water. Water is a fluid. Or think about your soda. That's a fluid. And think about how fluids move. If you were to shake your glass or shake water, the fluid inside moves pretty freely. And that's actually the case with these phospholipids. They're not set in a particular place and they can move actually. So they move actually with the consistency somewhat like oil. Think of oil you put in your car or cooking oil and how it moves slowly and sluggishly. And that's how these phospholipids move with each other. They kind of drift past each other really slowly. And that's important because that means the plasma membrane is somewhat flexible and can change shapes. Let's see what else is in the plasma membrane. So we discussed that there's phospholipids, but there's also a lot of membrane proteins. You notice that it's not only phospholipids, but you'll see these big purple structures in here. And these purple structures are some membrane proteins. So let's see what we have. First, we have peripheral protein. Peripheral proteins are such as these. They're basically on the outer edges of the bilayer. Just think what peripheral means. If you are driving and you say, hey, something's in your peripheral vision, what does that mean? It means they're somewhat like on the outside, right? So that's exactly what these peripheral proteins are. They're on the edges or they're on the outer side of the phospholipids. So it could be here, for instance, facing the actual outside environment, or here on this outer edge facing the inside of the cell. Next, we have integral or transmembrane proteins. These proteins, like the name says, they're integral, or integral means absolutely necessary, very important. So these integral proteins are integral to the actual membrane. In other words, notice they're embedded, or they go into these phospholipids. If you were to take out an integral protein, you would have these big holes in your bilayer, and they wouldn't function as well. So again, they're integral, or they're necessary for the membrane to stay in place. Whereas these peripheral proteins, if you were to pull off a peripheral protein, the whole membrane would still be intact. Now, the difference between a transmembrane protein and just a normal integral protein is that the transmembrane protein goes through the entire membrane. For example, this one I'm circling right here. Notice it goes from the outside all the way into the inside. So a transmembrane protein is a specific type of integral protein that goes through the entire membrane. Next, we have glycoprotein. So here's an example of a glycoprotein. The key feature about a glycoprotein is it has something called a glycocalyx attached to it. So see this green right here coming out? That is a glycocalyx. So notice it's attached to this glycoprotein. And that's actually going to be very important for identity. Now, what do we mean by identity? So we have trillions of cells in our body. It's important to be able to identify what type of cell it is from the outside. For example, let's say you have a sperm cell, and the sperm cell ultimately needs to find an egg cell. Now, from the outside, an egg cell is round. It probably looks like a lot of your other cells. So how does a sperm cell know that it needs to penetrate the egg cell and that it's actually found an egg cell and not just another round cell? That's where this glycoprotein with the glycocalyx comes into play. So this glycocalyx here is the identity flag, if you will. So each type of cell has a unique glycocalyx that says, hey, I'm an egg cell. So that way the sperm can come by and see that glycocalyx and therefore know that this belongs to an egg cell and that's the appropriate cell. Another thing that glycocalyxes are used for in their role as identity markers is actually, unfortunately in this case, HIV. So HIV is a virus, and what it does is it actually attaches to the outside of your plasma membrane and injects the viral material into the cell. And how it knows to actually attach to that particular cell, it attaches to your helper T cells, which are immune cells. It identifies that cell by the specific glycocalyx that your helper T immune cells have. So it'll target it, attach, and then again inject the viral genetic material into your cell. So actually one of the ways that researchers are trying to help prevent or cure HIV is to eliminate these glycocalyx markers on your helper T cells, so therefore the HIV virus has no way to identify them and infect them.